Good morning, everyone. I will not try to go into the discussion of in the definition. We can have several definitions, but at the end, the key point is if the product we have developed works or not works. Right? That's the point. What I like to, we know that nanotechnology and all the things you have discussed and have been presented in the last 2 days can alter the characteristic. I prefer to go with this definition myself of a drug, that is to increase its solubility, decrease degradation during circulation, and concentrate the drug at the desired site. The end result is improving the efficacy while decreasing avoided side effects. And I would like to stay with this definition of a nanodrug. And the development of effective, highly selective nanocarrier-based therapeutic modality has the potential to radically improve disease outcomes, although we have not a lot of successful stories up to now, especially when we go to the more complex nanodrugs we develop in academia and in the lab. Now, nanodrug components, and this is also an alternative definition, and I think Professor Gaspar mentioned about that nanodrugs will comprise several elements, and these elements include the biological active molecules, small molecules or peptides, macromolecular drugs, the encapsulated material, polymer, dendrimers, lipids, and organic materials, the surface coating in order to increase circulation of the nanocarrier in the system, in the human body, and of course the targeting moieties. If we put all these things together, we might come close to what we call a nanodrug, and I will not specify dimensions because experimental results quite often give different outcomes. Now, another definition, all these things are more or less different definition of the same, for the same thing. Now, these nano entities, being of the same molecular size as the biological entities, can readily interact with biomolecules sitting on the cell surface and within the cells. And this is also another alternative, I would say, definition of nanodrugs. Just to point out that a structure which is less than 20 nanometers can transit out of the blood vessels, and on the other hand, structure which is close to 100 nanometers can pass through the cell membrane via endocytosis. Things we have to consider in designing a nanodrug, in addition to the selection of the materials, of the drugs, of targeted moieties and so on, we have at the same time to consider issues related to the uptake and recycling, transendocytosis, and the somal escape. At the same time, we have to look at safety issue, in vitro and in vivo, cytotoxicity, hemocompatibility, immunogenicity, and so on. And at the same time, we have to deal with issues related to carrier biodistribution and degradation. Now, there are different carriers, nanocarriers, and I'm sure in different conferences in the last 2 days, you heard quite a lot about different types of liposomes, as you can see, molecular carriers, multifunctional dendrimers. If we look at the dimensions of all these type of different carriers, we can have sizes from 20 nanometers up to 200 nanometers, 300 nanometers, and we have results at least in vivo and animal experiments which are successful with all these different types of nanocarriers. Of course, the dimensions are quite different, and just to mention some of them. I will not go into the different types of nanodrugs or different types of nanocarriers we can synthesize or use for delivering a molecule, a biomolecule. Just I'd like to draw some, just to show you some results coming from our laboratory, because part of this presentation is production or synthesis of nanodrugs based on self-assembly. And what I'm presenting is a typical self-assembly process, starting with polyglycid or block polylactide blockopolymers we can obtain through self-assembly micelles. And these micelles, of course, they have a size of 30 nanometers. They carry negative zeta potential. In this particular case, have been loaded with oval albumin. They have been labeled for cell transfection, and these are the typical type of carriers we can obtain. On the other hand, we're producing this type of thiomer nanogels, and these are biodegradable nanogels, which can be obtained by cross-linking of thiol functionalized star polyethylene glycols or polyglycides in the reverse medium Olsen VH decimal addition with diacrylates. So starting, I don't know if I have a pointer, I must have a pointer. Okay, here. Okay, starting with this water in oil emulsion, in which in the water droplets we have dispersed the water-soluble hydrophilic polymers through this kind of cross-linking reaction, cross-linking the sulfide, cross-linking of hydrophilic polymers, processing hydroxylic groups with disulfide cross-linker. We can obtain different types of nanogels, which of course are not typical of 20 nanometers on 20 nanometers or 100 nanometers. They are in the size range of, as you can see, 300 nanometers. Of course, can be used for several applications. I will not continue with this type of systems you can produce in the lab, and I will move now to, I don't know how clearly you can see, for design nanodrug characteristics, and for the design, even myself I cannot see from here. They should be small, according to a recent publication by Ader et al. They should be small in the size of 20 to 200 nanometers. They should be able to encapsulate the active ingredient. Colloidal stable, so aggregation is a big issue, and these are some of the questions we like to discuss during the round table of we're going to have at the end of the session. They should have we should control the clearance mechanisms and the state of the nanodrugs. We have also to have what we call the triggering mechanism for releasing and even controlling the release of active compound in a desired way, just to mention some of the characteristics. Now, things which have come now into, we see more and more publications dealing with the effect of size on the fluid dynamics of the nanocarriers. And there are a lot of studies that the size can affect the fluid dynamics of the nanocarriers. And for a nanocarrier to be effective in delivering drugs to a diseased tissue, it should be able to efficiently interact with the capillary wall and migrate to the target tissue. Effect of size and particle internalization. There are a lot of results and studies on the surface properties and size of the nanodrugs in order to have an effective internalization of a nanodrug. Effect of shape now. There are a lot of results coming that the shape can have a critical role on the efficiency of a nanocarrier. The geometry, therefore, can play an important role in cell uptake of nanocarriers. And in this particular study, spherical and ovoid nanoparticles are more easily internalized compared to what we call elongated particles laying parallel to the cell membranes. There are studies and studies, but I will come with my issues and questions, which personally I'm not, we would like to see more systematic effort in putting together all the different results coming from different laboratories, and we need a system approach in order eventually to come with some predictive tools in designing this kind of nanocarriers. We have not seen these systematic efforts, and very often results are very conflicting, especially when we come to the size. For one study, 100 nanometers is an effective size. In other cases, it's not an effective, because, as we are going to see, it is far more difficult than the nanocarrier itself. Now, surface coating is also another important parameter we have to deal with in order to provide stealth properties to the nanodrugs, to the nanocarriers. And there are different types of polymers which have been used in order to increase circulation of the nanodrugs in the blood. We have these polyethylene glycols, hexamers and poloxamines, just to mention some of the surface coated materials which are used. And you know, all these things, if you measurement, for example, is an important thing. Do you measure the size of the nanodrug in the lab, under what conditions? And you decorate all these nanodrugs with different modalities, with polyethylene glycols. What is the effective diameter in this case? The nanocarrier itself might be 50 nanometers, but introducing a coating, a surface coating, the hydrodynamic radius can be twice or 3 times as big, as large as the size. So there are a number of things we have to consider. And up to now, we have not received very convincing and systematic answer. Now, targeting is another part of the nanodrug, and ligands grafted at the surface of nanocarriers can allow for active targeting in addition to passive targeting, which has been mentioned. And these ligands can actually bind to the receptors which are overexpressed in the case of cancer cells. Now, what is the mechanism of, and this is a cartoon, and I would like to be true, this cartoon, because everybody claims that his here nanodrug actually does this kind of, follows this kind of steps. Now, ideally, what we would like to have is our nanodrugs and nanocarriers marginate to the vessel wall and adhere to endothelium. So, designing even nanoparticles to do this kind of functionality is an extremely important issue, and I have not seen a general answer to this. Then, the second step is how the nanoparticles can penetrate the endothelium cells in order to reach the tissue. According to this cartoon, nanoparticles can release a reagent to break down the tight junction between the endothelium cells and the basement membranes. And then in the final stage, the nanoparticles interact with target cell membranes and then deliver the intended payload. What I'd like to know is what are the physics behind this, what are the times, and what the characteristic times dealing with all these different processes. And how much time we need for nanoparticles sitting on endothelial cells, how much time we need for crossing these endothelial cells or the basement membrane. What is the time which is required for all these particles sitting outside the cell membrane to penetrate, and what is the time of release of the payload to the cytoplasm? So these are important questions. I don't have all the answers. Now, what are the classical approaches based on the description which we receive? Up to now, We have used in our laboratories what we call self-assembly or emulsion-based methods. And these are classical, what we call bottom-up synthesis, starting with classical synthesis or nanoemulsions. We try to incorporate into these nanoemulsions or micelles and so on. We try to incorporate the active ingredient. This is the classical approach, and whatever has been presented and has been published so far in one way or another deals with this type of nanoparticles and nanocomplexes. There are a lot of questions. What is the size and how broad or narrow is the distribution? What is the concentration of the active ingredients with respect to the size of a polydispersed formulation? Because in the laboratory we produce this type of nanocarriers. They have a certain distribution. We can have nanocarriers from 20 nanometers up to, say, 100, and we like to know what is the concentration of the active component in the particles of different sizes. So the composition of the different spherical of the different particles is a big issue. On the other hand, there are questions related to the shape of the particles. Can we tune the shape of these particles? Recently, and I think Professor Gaspar in his previous presentation presented some new approaches which are based in top-down fabrication methods coming from the electronic industry. And what is the case is that this technique can be used for a long time from the electronic industry using nanolithography, as we're going to see, for producing nanoparticles of uniform size. And in the electronic industry there is a lot of discussion to use self-assembly methods in order to obtain nanoentities. But what we see now is that in the nanomedicine now, in the fabrication of nanodrugs, there are a lot of reports referring to top-down fabrication. And here I have a compilation of some of their techniques out of the wide variety of nanofabrication processes. Nano-imprint lithography, which is used again in the electronic industry, included step, and flash imprint lithography can result into this type of unisize nanoparticles. We can control the size, the shape, and the composition. And the same is true for the print type of technology. And there is also an alternative technology, what is called solvent molding based fabrication. So we see now developments using this top-down synthesis approaches which allow us to obtain shape-specific nano and microcarrier products. Now, what is a design map and what we have to reconciliate eventually? And this, I have 3 slides to close this brief presentation, and I actually to bring to your attention these thoughts. Personally, I have not received a satisfactory answer to all these questions which I will pose to you. For the design of an intravascularly injected nanocarrier in the bloodstream, 3 processes: margination, as we know, adhesion, cellular uptake, need to be analyzed as a function of the following things, of the nanovector design parameters: size, shape, surface properties, bulk properties, surface density of targeted moieties. All these parameters must be analyzed in a systematic way. Also in terms of the biological characteristics of the targeted tissue. And these biological characteristics: receptor density, blood flow, descriptors, wall permeability. This is one question we would like to see a systematic answer to this complex question. Now, another question which I like to pose is that what are the problems related with the crossing of different biological barriers? We know that most nanomedicines are currently administered parenterally, but both the market and patients would prefer other routes as well, pulmonary, nasal, dermal, and so on. We like to know what are the mechanisms and how we can achieve the crossing of different biological barriers. Here I have a cartoon for transepithelial pathways developed for transfer from intestinal lumen to the bloodstream. And as you know, for oral administration of nanodrugs, this is one of the most important issues. And we're expecting the results from second stage application exactly on this subject. I don't know if any of you is one of the evaluators of our proposal. Now, with conclusion and also with some recommendation, the fact that only a few nanodrugs, nanodrug formulation, have reached the market indicates that the successful transfer of the lab research results and also results from animal testing to the clinical application is a very difficult issue. And I don't know what is the answer personally. Apart from the small size of nanodrugs, we seek to satisfy in order to fulfill specific physicochemical criteria, biocompatibility, biodegradation, colloidal stability, protection of active therapeutic control release kinetics, things which I didn't mention, improve pharmacokinetics toxicity issue. And there is an extremely important issue, and there was 2 weeks ago in Brussels, there was a big conference on safety, toxicity, on toxicology, and these issues are going in parallel. We have also to have methods of producing these nanodrugs which are simple. The industry should produce this kind of nanodrugs in a cost-effective way, and the process must be scalable. This is not a trivial thing, to have a scale-up process. What is the suggestion is that we have to have a new paradigm. And the new paradigm is, as with the rational drug design, which was introduced within the pharmaceutical community decades ago, predictive mathematical models combined with in vivo assays and in vivo testing need to be integrated now in a very systematic and what I call computational type of approach in order to see what kind of rules and guidelines or what kind of lessons we can learn in order to come with what we call rational design of nanoparticulate systems. The things, therefore, we propose is what we call a system biology approach from a mathematical point of view, from a computational point of view, starting all the way from omics and going to the cell, to the tissue, to the organs, to come with a computational. It's very easy to show this cartoon. Might require 10-20 years work to come with some meaningful results. But in my opinion, we have to have an additional route to the classical route we have followed up to now, with a lot of successes, but with a lot of failure as well. And just to leave you from a recent publication of Tinkler, the computational approach for safe design, where we can pull now using what we call a computational platform, which combines computational tools, combines toxicological information, combines results from high throughput analysis, in order to come with what we call basic and clinical research, engineer nanoscale materials, implication and application, dose response, structure, activity, relationships, all these things, personally, I have not seen in a very systematic way. If it is the case, 10 years, this is probably my prediction to come with what we call computational modeling for safe design of nanodrugs. Thank you very much.